Let's have a quick tour of the brain, just some of the characteristics that make up this thing called the last frontier. This little sea squirt, Emperor Hirohito in the 1940s, when he wasn't otherwise engaged in making war, studied marine creatures, and this particular one spent the early part of its life swimming around. It had a tiny brain making decisions about what to eat and what to avoid and where to move to next and things like that. But it reaches a stage in its life cycle where it attaches itself to a rock and slowly eats its own brain. I guess confirming that our brain facilitates our interaction with the environment. It's an essential part of being engaged and getting our needs met from the environment. Some of the physical characteristics: the brain is encased in a fluid-filled sac inside a hard bone case. Our skull is not unlike an eggshell, and inside is our brain. And what's not generally understood is that if you took the human brain out of the skull and put it on the laboratory bench, it would just be similar in texture to a raw egg. In other words, it wouldn't hold its shape. The examples you see used in scientific experiments are mostly set by being immersed in formalin. Brain weighs about 1.3 kilograms, which is probably one of the explanations that people suffer neck pain so frequently, because of the weight that's being held on a fairly small collection of muscles. 23% of the body's oxygen. Oxygen, of course, is the fuel for the brain. The body can manage without water for about 10 days, food for up to about 40, but oxygen, it'll only last a few minutes if it's deprived of oxygen. The cerebral cortex is this wrinkled covering over the brain itself. It's a very tightly compacted network of neurons called the grey matter, and it's actually a layer about 3 millimeters thick, and if you laid it out it would make up about the size of 4 A4 pages. So it's quite an area. The reason that it's so wrinkled, of course, is that this is all compacted into the outer covering of the brain itself. The two hemispheres are completely separated, except for at the back of the hemispheres, down around this area here, there's what's called the corpus callosum, a very tightly woven network of neurons that connect the two hemispheres. Interestingly enough, people that suffer from epilepsy, this is the area that there's an intense activity. In fact, there's a lot of short-circuiting going on in this area. And some of the people that suffer extreme epilepsy, surgeons have actually separated the two hemispheres completely so that they no longer suffer epilepsy. But it's not a common procedure because of all the other problems that arise. People are left quite often very confused because there's no interaction between the two hemispheres. And one of the areas, interestingly enough, that presents serious problems is in humour. People that have undergone split brain procedures, they do laugh at jokes, but they have no idea what they're laughing at. It's without this connection between the two hemispheres, it's not possible to construct the very subtle, often very subtle nuances involved in humour. All brain activity is a result of electrical and chemical signals passing from one neuron to the next. People sometimes make the erroneous connection that the brain is like a computer. While there are similarities, there is a very important distinction, and that is the wiring in a computer is the complete network of wires. They begin and they end, and if they're not broken, the electrical current will go from one end to the other. The brain, however, doesn't have a wiring as such. It has a series of neurons and a gap, a neuron and a gap and another neuron. And while the transmission is taking place here, it is like an electrical impulse, but this connection here is actually a chemical one. So it's an electrochemical one. It's much more complicated than just a simple wiring that you have in a computer. If we're looking at a single neuron, probably broccoli is a handy way of describing it. A neuron is made up of, first of all, the cell body. This is the nucleus, and like all cells, it is the basic building block of organic life. So here we have a cell, and in addition from the cell we have these dendrites, these very tiny, tightly connected little connections all over the end of a series of fibers connecting the cell to these things called the dendrites. Now axons are not shown very well in this broccoli example. Axons are actually the connections from the cell to the dendrite, and often, not always, one of the unique things about the brain is that there are so many exceptions, so that as a general rule we can talk about neurons and their characteristics, but there are enormous differences between neurons. For example, the axon on some neurons is probably only about 1 millimeter long. If we're talking about a nerve cell that's in the sciatic nerve, for example, it goes from the base of the spine all the way down the leg, and it finishes in the toes, and it's about 1 meter long. So they are quite different, and in general rule the transmission is from the dendrites through the nerve cell, and it connects to the other neuron via the axon, but not always. Sometimes the transmission is going the other way. During the 8th month of gestation, a pruning of non-connected neurons takes place in the womb. It's actual pruning that reduces the number of neurons to about half, from 200 billion down to 100 billion. Quite an amazing process, in that if it's not a process that takes place properly, it leaves an excess of neurons. One of the characteristics of autism is believed to be a breakdown in this pruning process. Their hypersensitivity to touch and to hearing and to sight and things like that is explained because they don't just have a single neuronal pathway, they have a whole web of interconnected pathways that heighten. Their senses are in fact heightened, which is a very, very disturbing situation. So this pruning is changing the number of neurons, the ones that are not connected, pruning them down to about half. Now, interestingly enough, these pruned neurons, here's the dendrites, there's the brain cell, and then the axon continuing down there to its connections to other dendrites. This pruning, the cells that are no longer needed, in other words, they weren't adequately connected to other neurons, so they die and provide nutrient to the strengthening or the speeding up of the transmission of the neurons that did survive, and it makes what's called a myelin sheath around this axon. As I say, it speeds up the transmission. So this process of neurons that are not needed die off and provide nutrient, similar to what we saw in the sea squirt. Interesting, when I talked about the gaps between the neurons, they're not connected like a wire in a computer. The transmission of the impulse goes from the axon across this gap called the synaptic cleft or the synapse, and it's got a receptor, which is one of these ends of the broccoli, these dendrites. And that's why the whole thinking process is so extraordinarily complex, is that it's not just a known neural pathway. There's a number of factors that will determine whether that impulse to make up a thought or to make sense of something, whether that connection is made or whether it's not. Interestingly enough, this diagram's really talking about the function of SSRIs. The most common antidepressants are in this family of serotonin, selective serotonin reuptake inhibitors. That's what it stands for. And what it actually does is that in this synaptic cleft, serotonin, which is regarded as the feel-good chemical, is released. At the end of a big meal, you might pat your belly and say, "Oh, that was lovely, I really enjoyed that." Well, that feel-good sensation is the presence of these serotonin neurotransmitters in this gap. Now, normally they have a way in which they're reuptake. They're taken back from the synaptic cleft into wherever they're stored on this the end of this axon. Now, it's believed that if depression is a result of not enough of this feel-good chemical, that if we tampered with this neurotransmitter region and left it so that the reuptake was inhibited, in other words, it didn't take place, that the person would remain in this feel-good state. In other words, the serotonin wouldn't be taken back. Completely erroneous, as it's been since proven, but it hasn't altered the way SSRIs are still so widely prescribed and used, with the belief that it really does make a difference. Another interesting thing about the brain is that it assembles perception by simultaneous interaction of whole images. It does not assemble thoughts from bits of data. An example would be the way we perceive things. Very rarely would a person see this image here and interpret in terms of assembling what this black thing means connected to that black shape and this black shape and so on. It tends to be a simultaneous interaction. In other words, we see a collection of things, and most people can sort of attach meaning to it rather quickly, that it is in fact a white lettering of the words fly. One of the other interesting things that's been found recently is that by using monkeys, training them to do a manipulative task, getting food out of a screw-top jar in fact, while their brains are hooked up to various scanning equipment, found that the increased neuronal activity was taking place while learning. In fact, learning is the time when there is most neuronal activity, an intense attempt to make sense. So a learning task takes place in a certain part of the brain, and there is an enormous amount of energy directed to that. However, when the task is no longer unfamiliar, in other words, they know how to do it, the activity no longer continues to take place. It's shifted to another part of the brain called a lower order part to free up this part of the brain that is dedicated for this increased neuronal activity. In other words, a specific part of the brain is used for learning, and learnt material doesn't stay there. It goes somewhere else so that that part is free to be taking on new learning. Another interesting thing is that neurons gain their identity. They are not predetermined. One interesting example in Italy, a little boy he was blind in one eye, and they just couldn't quite work out how that was the case. As far as their investigations, Everything was right. The eyes take in the stimuli, and it's processed at the rear of the brain here, called the occipital lobe. And when the first few weeks of being born, there's a massive migration of neurons. In other words, they're reaching out and connecting to other neurons right across the cortex. And for example, the neuron pathway needs to be made between the eye, which is the receiving sense, and the processing, which is in the occipital lobe. So the neurons migrate and connect up and make this pathway. And it's made by what's called NGF, which is neuron growth factor, and it's caused by stimuli. In other words, what the baby is looking at and making sense of, there's a lot of activity in the sense organ, and it stimulates this nerve growth factor and increases the link up of these neurons that weren't predetermined to become vision neurons, but they become vision neurons as the stimuli is applied, and they make the connection between the occipital lobe and the eye. Now, getting back to this little boy in Italy, he had an eye infection, so when it was treated, it was covered for about a week, and unfortunately this migration was taking place during that week, and while this eye was covered, there was no stimuli, so no connection between his one eye and the occipital lobe, and it couldn't be later connected. In other words, that was the window of opportunity. It closed in that time, and a connection was never made between his eye and the processing facility. What it does highlight is the crucial importance of stimuli in the early stages. One of the factors that really raised the importance of this stimuli is the number of babies, or young people, many of them were, adopted into America and Canada from the Eastern Bloc countries. After the fall of the Berlin Wall, they opened up a lot of orphanages, and American people adopted them. And one of the unfortunate aspects was that a lot of the young people they had, although they were provided for very, very well, they had an inability to observe things, to notice things. Some of the exasperated families would take them to Aspen, Colorado, and they'd say to these young people, perhaps teenagers at this stage, "Have you any idea where we are? Can you just see the magnificence of this place?" And the young people would kind of look around, and they really wouldn't notice anything. It was as though they couldn't be affected by their environment. And one of the reasons for that is believed to be the lack of connection, the lack of neuronal pathways being formed at this crucial early stage. When I was teaching psychology, I used to encourage the young people to don't devalue the early mother going over the side of the bassinet going coochie 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 coo. Now this mother realises that the baby has a very, very limited understanding of what she says. Language understanding comes quite a bit later. But it doesn't stop her doing all sorts of things absolutely crucial for the baby's development, because it's providing a stimuli, forget about whether it makes sense or not, it's stimuli that develops this growth. The neurons are actually forming pathways. It has good connections, and it's waiting for specific assignments. And this lack of stimulation is crucial, especially in later life. Perception is more than sense organs capturing incoming stimulus. There should be an image here. I wonder. Here we go. Quite often the early development centres on characteristics of a face, baby's capacity to understand what this configuration, they look at the features such as the eyes and the nose. Maggie Thatcher, I hope she doesn't find this disrespectful. And they carry these features but it's really made sense by the configuration, how they're arranged, and it's amazing that that's part of our perception, and it stays. If, for example, we look at an image, now this image is the same face and it's just inverted, but we can still see that it's a face because of the configuration of those particular features of the eyes, the nose and the mouth. This particular image was doctored a little bit. Just the mouth changed and the eyes changed a little bit, the nose left as it was. It still passes as a representation, a pretty close representation of the original one. That's the way perception works. If, however, I reverse this particular image, the one that's been doctored, like rather grotesque, what we're suggesting is that it's really a matter of looking at local features and their configuration, their relation to one another, not the very, very subtle detail that obviously when you look at it in this way you can see is quite different. I like this definition. This is about cognition, really, the ability to synchronize electrical activity. When I'm talking to my students about the brain processing, it's important to understand that there's an incredibly complex web. It's not just a single processing that will happen every time in exactly the same way. It is what a thought is a very original, unique development. And cognition is this synchronizing of the electrical activity to make a single coherent thought, a connection between our brain and what finish up in language or understanding or memory. I really like John Ratey's definition. He talks about this process as a choir faintly humming their own favourite song. When clapped, the entire group bursts into coherent four-part harmony. I think it's interesting to see that the neurons are really just like a choir. They're online, they're partially engaged, but when they're called on for this electrical activity to be synchronized, in other words, to make a thought, it's like they really come together and the connection between all these different neurons become a coherent four-part harmony. In other words, a clear thought. Most of this material comes from Ratey's amazing book, A User's Guide to the Brain. Thank you for listening.
Made with Doodlecast Pro.